आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़।
We bring you all a podcast series on STEM popularization, that is, science, technology, engineering, mathematics, and medicine, supported and catalyzed by the National Council for Science and Technology Communication, the Government of India. Program is produced by Media for Community Foundation.
Welcome to another episode of the podcast from the Indo French Center for the Promotion of Advanced Research, CEFIPRA. Here, we highlight the groundbreaking collaborations between Indian and French scientists across diverse scientific fields. In this episode, we explore a project that's tackling a fundamental challenge in chemistry with potential implications for drug discovery and material science. This initiative, supported by CEFIPRA, unites the expertise of Professor Srinivas from Indian Institute of Science Education and Research, Pune, and Professor Thierry from the École Nationale Supérieure de Chimie de Rennes. The focus of their collaboration is glycolization, the process of attaching sugar molecules to another molecules. Glycolization plays a crucial role in many biological processes, but chemically, it's a very tricky reaction to control. Even over a century of research, there's still no universal method to create specific sugar linkages. This project aims to deepen our understanding of glycolization and develop new and more effective ways to synthesize complex carbohydrates. The researchers are exploring novel approaches, including investigating the mechanisms of new type of glycosyl donors and discovering new leaving groups to facilitate the process. They are also working on making these glycolization reactions more sustainable, efficient, and scalable using continuous flow processes. And now, let's listen from them.
I am Professor Srinivas Hotha from IIS here in Pune. I've been here for the last 15 years. I joined here in 2010. And before that, I was at NCL Pune as a senior scientist. And I returned from US in 2003 after a postdoctoral stint at the Rockefeller University in New York City. My lab specializes in making molecules which are sweet. For example, if you look at the common sugar that you look at on the table of any kind of a dining table, you realize that there is something called sucrose. Sucrose is nothing but a molecule which has got two sugars. One is glucose and the other one is fructose. And now the challenge is, let's say that this is glucose and this is fructose. And how do we make a bond between them so that we will be able to get a compound which is like sucrose? And this kind of thing has allured the mankind for a long period of time. And this bond was first made by Emil Fischer, which we call it as glycosidic bond. The bond that connects between a sugar and a non-sugar or a sugar is called as a glycosidic bond. How do we forge that bond is the most important thing in the field of organic chemistry. And while doing so, there are two chances. One is that either it can form the bond in this fashion or it can also form in this fashion too. So how do we forge this bond to make it in one of these two possible directions is the thing that is yet another important thing in the chemistry. So having said that, where all this is applicable is that one need not make the sucrose from chemistry because there is a lot of cane sugar that is available. But in a way, the nature uses these kinds of molecules to make something really good. So how does it use it is because suppose, for example, if I join more than one sugar, then it becomes like this. It could be a molecule which could be hanging from the actually the cell wall. Let's say that the cell is inside and all its components are protected by something called as a sugar chain, and this we call it as oligosaccharide. And this oligosaccharide, what it does is that it gives, and these are attached to something called lipids. Let's say this is the lipid molecule now, and it gets added, so that now what will happen is that we will be able to separate the cellular components from the external environment which is present, and thereby these molecules which are arrayed like this molecule plays an important role in cell-cell signaling and many other information transfer, and at the same time biological pathways. For example, in the case of Mycobacterium tuberculosis, the cell wall is very specific. It has some specific sugars and which are currently investigated for the discovery of drugs. So what ethambutol or isoniazide drug does is that it does not let this cell wall to form, and thereby it gets cut, and then the cell becomes vulnerable for the biological fluids, and hence what happens is that the cell death happens. The Mycobacterium tuberculosis dies. So these molecules play a phenomenal role. However, nobody understood, or very limited studies are there which actually tell you how this particular cell wall is forming, and if it has formed, then what is the role of the lipid that we have it here, and what is the terminal epitope that they are doing. They are all flexible. The molecule is quite flexible. And does it form any kind of a self-assembled structure or not? And if it is doing like that, then what should happen is these kinds of molecules and will become flexible, and they form self-assembled structures. And these self-assemblies, how do we study? So to study those things, what we require is a method of constructing this particular bond. The bond that forges the two sugar residues which are very flexible. And like this particular bond, so what we did is in our lab way back in 2006, with the funds from the CEFIPRA, DST and other funding agencies, we found a new way to forge this particular bond. So what we did essentially is that we were able to connect two sugar residues. How do we do that? It's a very thermodynamically unfavorable process, and by using some catalysts, if for example in our lab what we did is that we found a process by means of which one will be able to forge the bonds in this particular fashion. So what we did is we activated, so with the funds that are available from the CEFIPRA and other funding agencies, what we tried to do is we developed a process by means of which one can forge these two bonds. How do we do that one? Is that we use the catalysis, and the catalyst that we use are having the gold as its central atom. And now what the gold does is that it will actually activate the triple bond, which is nothing but a carbon with sp hybridization. And now what does it do? It activates the triple bond, because of which it leaves, and there is some appendage which will be leaving from here, and the bond gets forged. And by repetitive process we are able to forge these bonds. This is a particular art that we developed in my lab, and now we are at a position to make bonds which are as complex and as big as this particular molecule, which has got about 131 sugar residues which are placed next to each other, which is nothing but a phenomenal advancement in the field that was possible only because of the funding that we received from the CEFIPRA especially, and from other funding agencies. Okay, so now is it the only thing that we do in our lab to make this kind of molecules? It's not like that. So the same process can be utilized to make molecules such as this one, where you have a central carbohydrate or saccharide molecule, and on every side you have yet another one. And if you look at the intricacies of this particular molecule, now you have a variety of bonds which are present, and these actually create a lot of steric hindrance for the incoming bond to be placed. Yet another one cannot go. So because of that one, we have to play with the stereoelectronics of this molecule so that we will be able to assemble these molecules. Now, once we have these assembled molecules, now what can we do? So we can study how they actually form hierarchical structures. So for example, I show it like this, but it can also be like this, or any kind of a conformation that is possible. And these are all flexible bonds. But let's say that if I have a kind of interaction between this red sugar and this yellow sugar, then the molecule takes this space and it will be coming closer in space. But suppose instead of that one, let's say the blue one took part, and then we'll have yet another shape. Or if not, maybe this blue can have an interaction with this black one, then we'll get yet another shape. And it is the Mycobacterium tuberculosis takes or makes use of all such kind of bonds and interactions to make the cell wall a very unique one, so that now the organism can live in our lungs and other places for a long period of time. So that means that it will be living inside the body of human beings for a long period of time without actually making the symptoms of the disease actually show up. So now, if we can make a tool that exploits these interactions, then what will happen? We will be able to detect those microorganisms or Mycobacterium tuberculosis inside our lungs, though they have not actually shown up the disease characteristics. So thereby we will be able to save patients in a much better manner. With that intention, we proposed a project to the CEFIPRA where we thought that we will develop a diagnostic tool for Mycobacterium tuberculosis cell wall detection. And the far-fetching ideas and then these things are that by medicinal chemists, what one can do is that they can make probes that will enable them to detect Mycobacterium tuberculosis presence in otherwise inaccessible places of the human body. So one need not perform any surgery to detect Mycobacterium tuberculosis, but they shall be able to detect the tuberculi in the lungs of patients. So what we did, we made several analogs of these cell walls and then put them to the testing thing. Though the studies are still continuing, and at that point what we also have thought is that we will be able to shed light on how do we make these bonds. One of the things that we did is that we discovered yet another way to forge this bond by even easier methods. For example, one of the bonds that we did is that instead of putting a glycosidic bond, what we did is that we have put a carbonate donor so that the carbonates are much more stable and they become solids also when it is required. So some of the substituents which are present on the sugar molecule make them to become solids, and these solids, because they can be easily transported, so we have sent those samples to our French collaborators, and they have also performed some of the studies that are required to understand the bond formation, and they have elucidated the mechanism of this particular reaction. This particular tool has given us a great handle to assemble many more molecules in an easier and faster manner. So now, having made these molecules, and then the French team in the University of Caen, and I have also collaborated with yet another group in University of Montpellier. And these two groups are, one group is specialized in understanding the glycosidic bond formation and elucidating the mechanisms, whereas the other group in University of Montpellier is interested in development of novel catalytic systems for forging this kind of bonds. So during the first phase of first project that I did with Cefipra, so what we have understood is that by making subtle changes to the reaction conditions, our abilities to increase the forging, increase the number of bonds that we can forge got increased, and thereby we have reported several compounds with more than 20 residues being added to each other. Okay, for example, at that particular point of time, the world record was something like some number, and we have also set a new world record in the number of glycosidic bonds that we could make in a chemical flask such as round bottom flask, and we held that particular record for certain amount of time before we lost our record to the Chinese counterparts. But however, the issue that happens is that from India, we have pushed the boundaries of glycosines by forging as many more bonds, and we have made the longest oligosaccharide ever synthesized on Indian soil. So having said that, now what is the next thing? So what we thought was, so there are some problems which are encountered during the process of assembling these glycosides. So the yields were poor. So in an effort to increase the efficiency of the glycosylation, what we found is that by putting the carbonate, one will be able to conduct these reactions in a very easy and mild manner. So thereby our number of residues that we can forge got increased phenomenally per year, and we could now conveniently have developed processes in such a way that we can achieve not 20 sugars, but at least the longest one that we made is about 54 sugar residues which are forged next to each other. Why did we do that? Because as I said, these are all part of the Mycobacterium tuberculosis cell wall. And then we spent quite a lot of time to understand their significance in the by using various biophysical tools, and we are at a stage to understand that now whether the molecule, okay, now let's say these two sugars are attached like this. What if the sugar two residues are attached like this? What kind of self-assembled properties it will show? Or it can also get attached like this. I mean, how does it change the course of the structure that it forms? And nature, or Mycobacterium tuberculosis, takes one of these, not the one that I showed you, but it uses in this fashion. So why did it choose this particular orientation over this particular orientation is a question that we are trying to address. And by doing so, what will happen is that we will be able to contribute to the overall understanding of the pathogenicity of the molecules, and ultimately with a far-reaching goal that if I can inhibit the process of making this particular one, then that could become very specific for Mycobacterium tuberculosis, thereby it could become a potential drug. So now let's say that if I find this molecule which can intercept this formation of this particular unit here, then essentially what will happen is that this particular thing can be used as a drug for curing the disease. In the second Cefipra project with the University of Montpellier, so what we have done is that we developed reaction conditions to facilitate in an easier manner using earth-abundant metals. So for example, in the first Cefipra project, we discovered that we have used gold catalyzed glycosylation process. Now what we are trying to do in the second Cefipra project is that we are trying to make these bonds, but now instead of gold we are trying to use other metals or light as a medium to forge this bond, so that we will be able to conduct reactions in a sustainable manner. The group led by Florian and Mark Telfer in the University of Montpellier has earlier developed a lot of processes for chemical reactions in a sustainable manner. So we are collaborating with them to develop such kind of reactions and understand their mechanism by using mass spec and other tools. So during the course of CEFIPRA project, it gave me an opportunity to visit France a couple of times, and once to the University of Rennes, and once I went personally to the University of Montpellier. And by going there and then seeing the kind of facilities they have and the infrastructure that is there in those institutions, and the kind of work ethics they follow there, and the interactions during the visit are phenomenal, because that actually contributed a lot to the overall networking events that are organized during the process of the visit are really interesting, because that is the way we can understand the culture and respect each other for the kind of ideas that we bring to the table, and thereby we could actually improve upon some of the research ideas that we have. And this CEFIPRA is the only project funding that gives funds for consumables, equipment, and student visits as well. So for a PI to go there and then see their infrastructure and discuss our ideas with them, and then get contribution from them and contribute to their research ideas is a very good thing. But for the student who is in the nascent stages of development of their career, it's highly important. And the couple of students who went from our lab, my lab, to the University of Rennes and the University of Montpellier, they came back fully charged, and their ability to think independently got improved phenomenally, because they saw what kind of facilities exist in the developed countries, and at the same time in France especially, how the major discoveries happen because of the free thinking that is possible, and at the same time they could interact with those experts who were there in the field, and that has contributed a significant jump to their overall career. For example, the first student, after that completion of the PhD, now he is actually doing a postdoc in US, and then he is going to establish his own research group very soon in India. And the second student who went from my lab to the University of Montpellier is currently working in the American Chemical Society as the editorial assistant. And when I talked to her before and after visiting France, her ideas about science have got clarity, and she could then make very good impactful research presentations, and she could also clearly express herself about the kind of research that she did over there in France for 1 month. And I think this is the only funding mechanism in India that exists for that enables students' visits. And I'm indeed grateful to the research funding from CEFIPRA, because getting 6 years of continuous funding from CEFIPRA, and during very testing times of the COVID. So we started the project at a stage when COVID was about to set into the world, and CEFIPRA, being CEFIPRA, it has actually committed itself to make researchers empower with funds, and then think freely, and then work freely, collaborate freely, and contribute to the cause. And I really have no words to express myself the contribution that CEFIPRA gave to me and my lab, because of which we could do meaningful research, and we could at least develop a protocol for forging bonds.
The findings from this project will be shared through publication in top international journals, and where appropriate outcome through patents, potentially leading to new industrial collaborations. This is Harsha from CEFIPRA, signing off till we meet again with another project with another scientist.
आइए हम सुने विज्ञान की अनसुने